Good morning, good morning, good morning. It's Friday. You made it. It's the end of the week. Caramel says, congratulations. And I don't know if you can hear her purring. I hope you can. It's one of my favorite sounds in the world. And today we are going to be talking about some of your favorite taxa in the world. The Cnidarians. Hooray! Everything jellies all the time. Smith continues to check the waiting room. People are coming in. All right, I'm going to thank you for finding all of those. That's very, very good. There's a couple more that are stored in the, oh my God. There's a couple more that are stored in the PDF when you get that. All right, clear all the drawings. Let's start moving forward from now. Do jellyfish count as an invertebrate? They certainly do. This is the Cnidaria that we're going to be talking about today. This is a view of a absolutely stunning Cnidaria that the Sebastian ROV filmed at the bottom about 1,000 meters, 911 meters underneath the Antarctic ice. This is a feeding Cnidaria, and you can see lots of the arms and the mouth and the center, and oh my God, it's stunning. And if you haven't already, I've been posting some links on the welcome page for your course link. There are these dives that are being streamed and conducted right now. This is from yesterday. Put in the chat, this is a animal. What phylum is this animal? This is Antarctic fauna. This is not an emperor penguin, but oh my goodness, this is gorgeous. Give me a couple of best guesses. What is this hairy wonder? Is it a jelly? Is it a ketognath? Is it a slug? That's a guess. So is it a mollusk? Sure looks like it could be a mollusk. That's actually a really good guess. A fluffy sea slug, a nudibranch. Good guesses. These are all good guesses. A sea bunny. Yeah, Gavin, polychaete. So this is an annelid. And what's happening here, they saw this gorgeous annelid and they scooped it up. This is their slurp gun. And you've seen some of these in action before. You'll see some more. This is one of the really kind of gentle ways of sampling things that as we move away from dredges, just kind of drawing across the bottom of the ocean, this is a subtle way both to see the animal in its natural habitat, to make observations. Remember in your first lab with your notebook, like observations are absolutely the key to understanding the natural world. So it's an annelid. So a polychaete is an old name for a group within the annelids. A scale worm, and the exact family is likely something we called Aphroditeae. Slurp gun gets vacuumed. You got it. And this was this morning. Like they're bumping into them again. This is a few hundred meters higher, but here it is. It seems in a all-out death race between the polychaete and the sea cucumber there. So watch these videos. You can get, if you click on that image later on, you can go exactly to the stored files from the end of that dive. You can watch them live, or you can watch all of the liveness recorded. Yay to that. All right, so where were we on Wednesday? We were talking about a whole bunch of things. We hit 3 different invertebrate phyla. We talked about sponges and how they're not boring and how they are predators and how they reproduce. We talked about the mighty, mighty placozoans, and we listened to Soul Bossa Nova, and we talked about how there's many more groups than we historically thought. And then we began our journey into the Cnidaria, and we talked about some synapomorphies for this group, characteristics, the strobulating, and that's the image that you see here where the polypoid form gets a little kind of like an elastic band around it and it births off, asexually squeezes off a medusoid form. And I suggested that strobulating Cnidarians might be a challenger for some of the cutest things in the known world. And some of you probably agree with me, because this is a ranking of the feedback we got in the Google document about which phylum does your favorite species sit in? And so many of you came up with Cnidaria, which has surprised me a little bit because I thought a bunch of mollusks are often first or second. And then I saw this, and I wondered if maybe those of you who are voting for the cephalochordates, which is like lancets and amphioxus and a kind of a small clade in our neighborhood in the deuterostomes, it's a beautiful day in that neighborhood, as it is in mine. I hope it is in yours. But I wondered if some of you were actually voting for cephalopods. But if I've got like a whole cohort of like a dozen people here who are passionate about cephalochordates, please put that in the chat, because I need to know that to like pump up the cephalochordate part. Not just the jam that's being pumped up, but the cephalochordate part. Oh my God, old Gen X jokes just like dropping from the sky. Today, we're going to talk about cnidarians. We're going to talk about their ecology, who eats them, who do they eat, about how they move, comparing and contrasting with the ctenophores. We'll talk about the diversity and talk about different, like a classic Tribiani group that I was taught was a real thing. And it's not. The cubozoids, the scyphozoans, the hydrozoans, which include the siphonophores, like the Portuguese man-o'-war, which is kind of a crazy, gorgeous, oh, conch on earth. Yes. Tip of the hat. Well done. And then we'll talk about a really odd lineage of cnidarian called the myxozoans. But we got to start, because we haven't started yet. This has all just been like excitement as we go. Come on in, find a seat. Let's begin. I showed you a video on Wednesday of strobolating, and I was wondering if that was going to be the cutest video, and I thought, oh, maybe there's a better, cuter video, and so here it is, strobolating, reproducing medusoid forms. That's awesome. So let's talk about ecology. To start out talking about how cnidarians affect ecology, and we're going to talk about it, remember with toxoplasma and even with the dinoflagellates, we talked about how a lot of marine animals are being negatively affected, adversely affected, or their ecosystem and their food web is being harmfully affected by terrestrial-based human processes, like increased production. So we've got lots and lots of different kind of elements that we apply that to the surface to grow our food, and they wash out into the ocean, and that causes a whole bunch of eutrophication there in estuaries and coastal lagoons. And so there's much more food that are available for opportunistic feeders, for things such as dinoflagellates we talked about with the protozoans, but also with these jellies. And we're seeing across the world many more cases, many more incidences of blooms, jellyfish blooms, which might sound like a lovely thing that you would hand to a partner on a gorgeous day of celebration. But a jellyfish bloom is actually really harmful because they can outcompete fish, and they have a very high resistance to levels of oxygen in the water that would preclude fish. And so once the fish are gone, it's very hard to get them back. So alterations in river discharge, damming, and also phosphorus and other phosphates and all sorts of fertilizer applications are changing that. And blooms change the distribution in the carbon and can dramatically affect economic values. And that's happening all over the world right now. This is not a kind of, oh, it's happened once or it's happening vaguely. It's like this is a newspaper article from yesterday in Tasmania, in Hobart, where there is a large jellyfish bloom going on very close to home there that the researchers nearby have blamed on an oversupply of nutrients in the water that have to do, in this case, with salmon aquaculture. Other crazy things about, so blooming, so numbers that we wouldn't have historically appreciated. How about jellyfish appearing in places that we wouldn't have historically appreciated, like freshwater lakes in Ontario, British Columbia, and other parts of Canada. There are invasive jellyfish in freshwater Canadian lakes. In particular, this jelly here, which is Craspedacusta, sowerby, Craspedacusta. That's another offered up swear word for you. And freshwater researchers, limnologists are concerned across the country because really we haven't yet studied what kind of effect this is going to have on the native species with which it now co-occurs for space and for food. This picture and this figure over here are from a recently published journal article in the Canadian Journal of Zoology that looks at, kind of graphs out for you the near exponential rise in the number of sightings, and these are based on iNaturalist sightings, citizen science informing the spread of this invasive jellyfish. So bloomin' jellyfish, invading jellyfish, stinging jellyfish, strobulating jellies. And that makes me think, well, what is the last thing a jellyfish might see? Many of you, as aficionados of the natural world, of marine and freshwater biology, probably know it's this source of terror. I'm not sure if you know what this is. Put in the chat if you're quick, if you got like loose fingers, tell me if you know what that is that we're looking at. Yeah, exactly, Joss. It is a mouth of a sea turtle. It puts that part in Finding Nemo with like, hey, dude, that kind of cool surfer turtle in a different light if you see it from the perspective of the poor jelly. Hey, dude, I will now eat you. So the last thing the jellyfish eat, so lots of things. We knew we've made observations like this. This is a big animal eating a big jelly. So in this case, we know, and we thought it was actually kind of a weird thing to see because historically, we're not sure a lot of the time of the things that eat jellies. However, so we talked about one of the couple of the threads of the currents that are going through this river of a course, one of them being you're living in an age of discovery. And it precedes you, but it's like exploding as you become a biologist because of the information that remotely operated vehicles are bringing back and other technology are bringing back from the natural world, and the sequencing technology that lets us look indirectly at who's related to whom and who's seeing whom, or who's eating whom in this case. And so what eats cnidarians? More than just terrifying sea turtles, more things than we thought. So using sampling essentially the gut contents, these researchers figured out there's about 50 different species of predators that are eating cnidarians, and very few of them did we know beforehand. So much to be learned, much to be learned. Another thread that is proceeding through the course. There's caramel. Let me scroll up, see if I missed any. How did they get to the lakes in the first place? So you're in the same, like it's not often intentional. It could be from the aquarium trade. That could be it. It could be with bait. It could be it could be boats, ballast water in some bigger lakes like the Great Lakes, that kind of thing. But it's almost certainly inadvertent. Yeah, I agree. I never knew, and I would like to see them as well. Conch on Earth, making a. I have to bring that in. Yeah, moon jelly. There you go, the moon jellyfish that are blooming out there in Hobart. Yeah, so how does the sea turtle eat? How do they know other predators eat jellyfish without getting stung? So they're sequencing the gut contents with cnidarian-specific primers, so they will amplify cnidarian DNA from the guts of all of those other animals. And so they're kind of compiling all of that information by sequencing the guts. Instead of looking through the guts, you can understand there's enormous bias when you search through gut contents to physically look to see who has eaten what. You have a bias towards arthropods, towards things that or mollusks, things that have a hard bit that generally tends to survive some sorts of digestion. If you're a squishy thing, even a squishy venomous thing like a cnidarian, or a squishy sticky thing like a ctenophore, we have a bias against finding out who eats you because we don't have good methods to look at that until we have DNA, when we can sequence your gut contents. All right, caramel. So we're going to kind of park the ecology for a second. We're going to go back a bit to the developmental biology and then into some of the movement. We'll talk about cnidarian development. And I'm going to ask you to draw on your memory of Wednesday when we talked about placozoans, because I made mention to this, but here we've got a figure. This is a typical solid hydrozoan, so a planular larva that results from the blastopore kind of ingressing. So we have a planular larva, meaning we have an endodermis and an ectodermis. And just with this really simple kind of larval stage, I hope that what you're seeing there is maybe there's some evolutionary similarity from the placozoan to the larval stage, this larval stage of the cnidarian. So the endodermis is going to become the gastrodermis, and this is a classic diploblast. Two germ layers, endo and ectodermis, but there's no mesodermis. As soon as we start to see a mesodermis, we're talking about a triploblast, and that's when we start to see through guts, and the frequency with which we'll talk about anuses and pooping will just go through the roof, and also excretion. But diploblasts generally kind of get rid of toxic byproducts of metabolism and life by kind of diffusion across their body wall. And remember how we looked at that classic line drawing of the sponge wall? That was from the classic text. There's a reference to the equivalent diagram in your new text here. I like the line drawing here for all the different cell types that are shown in this really beautiful line drawing of the cnidarian. So cross section, or a chunk, a cube. Some of you were talking about cubes earlier. The cubes of the cubosome. So we have cnidocytes, or nematocysts. We have muscles, and we have muscles that lie at right angles. So round and longitudinal muscles. So here's we've got a longitudinal muscle. Here we've got a circular muscle, and you can see there what I say at right angles. So the circular muscles are kind of going around the bell, and the long muscles are going over the top, but they are at right angles, like a 90 degree to each other. That becomes important when we start talking about movement. But then there's all sorts of other there's bits of cilia, there's glandular bits, but we've got a radially symmetrical body plan. And so symmetry, we haven't really touched on a lot of symmetry yet, right? Because the placozoans lack symmetry. The poriferans, the sponges, lack symmetry. This is our first radially symmetrical. This is the first symmetrical organism. When we get into the ctenophores, we'll talk about biradial symmetry, which is like radially symmetrical, but in two kind of angles. And we'll unpack symmetry, and then bilateral symmetry, which is, of course, us. We'll unpack more of that on Monday. Now, one of the classic adaptations of the cnidaria is that they live as colonies, and that even though lots of times when we think of the cnidaria, we think of this kind of classic boop, 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 mouth backwards swimming medusoid forms, it's the polypoid form. So think of the little anemone, but more. So polypoids do look like that's kind of an anemone or a coral, but in many, many lineages, and you'll see some examples today, there are so many different kind of jobs that these polypoid forms. And so thus the success of living colonially is that you can give different jobs to, just like the cell, like the diversity of metazoans was permitted by organelles, and so specialization of task, that is true with kind of scaling up to the colony of individuals living together, where you can have gastrozoids, things that feed, dactylozoids, gonozoids, that their job is producing the gametes, and all sorts of different spines. And so you'll see a picture of this one later on, because I love this one. This is a hydrozoan. It's a colonial hydrozoan, and the only place in the world, this is all of it, we find it living on the backs of mollusk shells that have been discarded by the mollusk and are lived in currently by hermit crabs. What kind of weird, that's a 3 phylum, so we've got cnidarians, mollusks, and arthropods involved, and then there's so many questions that we don't know about the Hydractinia, and so I just throw that out there to you to say, like, this is Smith saying, wow, figure it out, come back and teach me about it, so when you teach this course, you can teach others about it. Now, how do they, thinking about polypoids, thinking about all the times we've already talked about corals. We talked about corals when we talked about dinoflagellates and apicomplexans, these kind of symbiotic co-partners in that relationship. And we talked about the importance, even back in the first lecture, about cnidarians, the corals, the anthozoans, building ecosystems, right? Reefs. And so to do that, you need a skeleton. And there's different ways that different cnidarian species construct skeletons. So there's chitin. So hydroids, some of the ones pictured here, they have a chitinous periderm, an outer covering of chitin. There's calcium carbonate, like the brain coral, so sequestering that from the environment that they're living in. There are different sea plumes and other corals that create spicules and tall, kind of branching, very plant-like looking things. Then we have cnidarians where the static the skeleton is a hydrostatic skeleton, which we'll talk about more and more, especially as we get into the bilateria. So the hydrostatic skeleton, where there is a fluid-filled cavity surrounded by muscles, and especially muscles set at 90 degrees, where squeezing in one way or the other causes the redistribution of the fluid inside of that cavity. And then, of course, we have the fact that there's all sorts of foreign material. There's the zooanthellae and the zoocorelli, or the dinoflagellates, the apicomplexans, all these very species-specific symbiotic partners that live together. Now, the polypoid form, whether it's permitted to grow up off the bottom by a hydrostatic, a spicule, a calcium carbonate, or a chitinous skeleton, the polypoid form, if you were to slice it sagittally, looks kind of like this. We're expanding on the muscles now, the circular, and so these antagonistic. That's maybe a simpler way of me saying like right angles, because probably we didn't ask you to, so you probably don't have your compass or your protractor here. But antagonistic. So the epidermis is longitudinal muscle, and the gastrodermis is circular muscle. And so these antagonistic muscles form. So we've got contraction here. We've got, looking at the kind of the bottom diagram here, right in the center with the arrows going up and down, you can see there is a contraction of the coronal muscle. The corona is the circular muscle that goes around the outside, and that causes the bell to squeeze, which takes the liquid that's inside of this bell of the medusoid and pushes it out. So it essentially becomes jet propulsed through the water. Now, the epidermis, the longitudinal, that's kind of emphasizing the importance of the circular muscle to the free-living pelagic medusoid form. What about the polypoid that we just talked about being so important? Well, there's still some contraction that's important of the circular muscle, but the longitudinal muscle becomes very important to the polypoid, because what it does is help you shrink away from nibbly fish that want to come down and feed upon you. And to do all of these things, to fire the muscles, the circular or the longitudinal, you need a nervous system. And this is one of the first times that we're bumping into a coordinated nervous system. So there is a nerve net, a diffuse nerve net, interneurons, no brain, but there are ganglia, which is kind of these concentrations, but nerve rings with concentrations of neurons that innervate the coronal muscle. And so you can see here, we've got the nerve net, not Skynet, this is not to be feared, but the nerve net that then surrounds all of that portion of the bell. And this is incredibly important for the polypoid to escape predation, to extend itself out into the water to feed, and for the medusoid form to move itself through the water to escape predation and to feed. So there's the inner nerve ring and there's the outer nerve ring. Now, when we put all of these things together, and I've already drawn my kind of hand puppet of how these things are moving. Here's a better view of one of the Ripley's Cnidarians. But I've got some questions for you. So we have the firing. We've got the elastic mesoglia. That's the jelly, right, of the jellyfish. And so we have an open form here on the bottom left. Then the firing of the contracted coronal muscle jets the water out. And so it moves forward. So it goes pulse forward. And so my question for you, there's the relaxed, is what causes the bell of the medusoid to refill? And so what I'd like you to do is to watch this and vote in the next kind of, 30 seconds here. You got some options. Is it the relaxed coronal muscle? Is it a contracted coronal muscle? Is it a mesoglia elasticity, relaxed longitudinal, or a contracted longitudinal muscle? And as you're answering, and use your annotate to kind of place your vote in one of these categories. And I will scroll through the chat for a second. Oh, good question. Why are some species symmetrical and some not? Yeah, we're going to talk about that a bunch on Monday. Promise. Or Wednesday, next week. Yeah, just a bunch of little guys standing together. Oh, to be a brainless creature, no anxiety, no homework, just waves. Awesome. All right. Take a sip of tea, and then remember, these are the consequence-free, low-risk great ways for you to be wrong here, right? Be wrong together and then learn from it. Okay, that's a very nice tea. Okay, thank you. Now, let's take a look. I'm going to clear those. Last chance to get in. So the common seems to be that, so the bell is emptied, jet propulsion by the firing of the coronal muscle, and then a relaxing of the longitudinal muscle is the hypothesis as to why that can be refilled. Let's take a look at what actually is happening. It's actually the mesoglia. So it's kind of passive. So there's an active pulse. The medusoid form, the bell opens up because as soon as the coronal muscle relaxes, the mesoglia, the jelly, wants to fill that space where it once was. And so it's that elastic rebound of the gelatinous jellyfish that causes it to go back to its original shape and to fill up with water again. Now, there's a new picture of gorgeous caramel, and I see no current ones. So we're going to switch gears and talk for the rest of our time together about some of the amazing groups here in the diversity. And just a word about this phylogeny of the major lineages of cnidarians is that if I was going to ask you about any of these, I would give you the phylogeny, and I would plot onto it some of the details, as are done here. So the Medusazoa, which is this group here, Medusazoa, that comprises some classic ones. And it's got Scyphozoans, Cubozoans, and Hydrozoans. And some of the synapomorphies for this group seem to be, or we thought were Medusoid stages, linear mitochondrial DNA. But you can see here really where we place the polypzoidea, and there's uncertainty as to whether or not this lineage is actually a monophyletic lineage. It all depends on where this sits and whether we include it with these synapomorphies. But it does include, monophyletic or not, the three that sit under it or within it, as we understand it now, are the kind of the largest lineages, the three major lineages. So the Anthozoans, it has these three, the Scyphozoans, the Cubozoans, and the Hydrozoans. When we talk about the three major lineages of Cnidaria, we're going to add to that group the Anthozoans, the corals, the mobile medusoid form, so the Medusazoa, and then the Mixozoa, which is a parasitic group, and we don't quite know where it sits. Stay tuned. And we talked about this on Wednesday. We talked about this life cycle. So we've got the Medusa, an alternating life history stage between polypoid and medusoid form, except in the Anthozoans that cycle, the polyps release the gametes, and we just have the polypoid form. So one of the first lineages we're going to talk about are the Scyphozoa, okay, which are characterized in this last by strobilation. So the budding off of that you saw earlier. So some of the cutest things. And there's an image of some gorgeous Scyphozoans. Synapomorphies or characteristics of the Scyphozoans include medusa with this new word to you, a sense organ called rhoeplia. Now, the rhoeplia are sense organs. And so they're acting as a bit of a statocyst, responding to gravity. There are some of them that can be adapted to seeing, not seeing, but sensing light and responding to light. And so when you imagine the medusoid form, left and right is not as important as up and down. So responding to gravity and responding to light when your life is in the water column are very important. So they have gastrodermal and epidermal components, and they're the elastic mesoglia. This is kind of a very characteristic jelly like we've been talking about as it moves through the water. And then they've got the statocysts, mechanophotoreceptors around the rhoeplium. Now, one of the other cool things, if you were to look at them in cross-section here, like this moon, is that we have gastric filaments. Okay? There's the eyeball, and there's the gastric filaments. The gastric filaments have both nematocysts to sting and calm the prey, and then the digestive cells to begin to eat the prey. Now, the strobilation, we talked about strobilation before because it's gorgeous, but here's the diagram from your text about it, that we have the polypoid that begins to pinch off, and then the aphyra, which is the juvenile or the young medusoid form that goes out, releases gametes that meet, bump into each other, form the planular larva that looks very suspiciously like a placozoan that settles and immediately changes form into the polypoid form. So there's the strobilation that's happening. All right. After the scyphozoa, I've got another check-in with Caramel. I don't see any new questions here. So Caramel and I will keep going. And we'll talk about the cubozoans. So this is another medusozoan lineage where the jelly is, and I think maybe I caught somebody who's talking about box-like or Minecrafty things in the chat earlier on, is that we have the cubozoans where they are radially symmetrical, but their bell is made into a box-like form. And we'll take a look at that. They have a lens replier eye, and this is a real specialization towards light. So it does have a lens for concentrating light and a velarium. Whoa, ho, ho. And I'll tell you about those kinds of things in a second. Now, they have small polyps that lack septa, lack digestive kind of branches, as you would see in the anthozoans. And this is a high-resolution look at a very small polypoid form, but a reduced polypoid stage. Really, it's all about the medusoids form stage here. So lacking septa means really it's not doing a lot of digesting. But you can see the Q, like with the kind of four tentacles, arms coming off, that's a real box-like shape, a cubic, and the four arms are called pedalia at four corners of the bell, and that's them there. And so we've also got these ocelli, or eyes, that are appearing here. And that's kind of amazing. We'll unpack, if you stick around for 3700 after this, if you're like, go away for the summer and you come back, I want more invertebrate enthusiasm. In 3700, we'll unpack vision across all of the invertebrate lineages, and we'll spend some time with the cubozoans and their lensed eyes, and a thing that has no brain and yet an eye, which is kind of amazing. All right, Caramel again, because we're going to move into, for the two more lineages before the end of our time together. One of them is the hydrozoa. And I love this one. So this is the gamete forming tissue is no longer kind of on that gastrodermis, it's on the epidermis. There's a loss of gut mesentery, so a simplification of where things are being consumed, and a simplification of the middle layer of the mesoglia. Now, this means there's a whole bunch of either adaptations into differentiation away from the classic medusoid form and away from the simplified polypoid form. And so this is a lineage where we do see classic medusoid and classic polypoid form. We see a lot of colonial forms here in the hydrozoa. This was where that commensal one with all the gastrozooids and dactylozoids and gonozoids and spines living on the back of the abandoned mollusk shell currently inhabited by the crab. This is the lineage that they sit within here. So when they do have a medusoid form, although the amount of mesoglia might be reduced, they have this velum. So the velum is this constriction on the inside of, so I'll do my bell again, and it's basically a lining on the inside. So comparatively less mesoglia, less elastic mesoglia. What the velum seems to do is it concentrates, it makes a smaller opening. So this finger is the velum sitting inside the bell, and when the bell squeezes, the velum reduces the amount of surface area that volume of water has to go through, which means it can move very precisely and sometimes very quickly. So in addition to the velum, they have endodermal cnidocytes that are absent, but then they have a lot of specialization across the rest of the body. They have gastrozooids that are feeding. So this is essentially, in this case, what you're looking at, the Physalia physalis, this is a colony. This is a floating colony. So it's not a coral colony building calcium carbonate structure on the bottom. It's not a commensal hydrozoan colony living on the back. of a shell. This is a floating colony that has gastrozooids for feeding, gonozooids for reproduction, and dactylozooids for defense and for capture of the prey. This is a diagram that I've lifted from a paper that you can go and look at the QR code or the URL, the DOI at the bottom. This is another output of Casey Dunn of the Dunn et al. Phylogeny. And I mentioned that he is a Cnidarian biologist, and he's particularly passionate about gelatinous siphonophores and these kinds of things. It's his analogy that the aliens crashing into the Earth are more than likely to land in the water, in the marine system, and if they're in the marine system, this is one of the first life forms of our planet that they're likely to bump into. And look at the different kind of jobs that are done here across the top. So this is what you would see if you've been, throw me in the chat if you've seen these kinds of things, but a gas-filled float playing a role in positive buoyancy and literally used as a sail to catch the wind and to move through the water column. So again, departures from the norm. And we talked about the medusoid form, and I said really it's about up and down with the Ropalia and the pedal eyes in the cubozoans and the other lineages, the scyphozoans. But here, left and right matter because they're kind of moving their colony through the water. And that needs there to be a pneumatophore or this gas-filled float. Then we have right up underneath the surface the gastrozooids. And so this is a polyp that ingests foods and carries out extracellular digestion of prey. No tentacles. It's about digesting the food, breaking it down, and sharing the byproducts of that digestion amongst the rest of the colony. Then we have what's called tentacular palpons. So this is a polyp, a tentacle-bearing polyp. That is unique to the Physalia physalis. It's used to capture prey and immobilize them with nematocysts. Then we have the gonodendrons, where we see all sorts of polypoid structures, some of which, like the jelly polyp, that we don't know what they do yet. Secondary accessory digestion, medusoid form potentially involved in locomotion, gonophore is involved in the production of gametes. So the pneumatophore, I'm just glanced down. Yes, it is, Brooke. It's essentially a swim bladder, and it's about keeping it kind of right up there on the surface. And that top bit, so this is the bit that would be filled with air. This bit sticks up and literally acts like a sail. That's kind of amazing. And so just a nod back, this is also a hydrozoan, another colonial hydrozoan that's living with its buddies on the back of the hermit crab shell, the abandoned mollusk shell. Now different zooids within the siphonophores, so we can see there's all sorts of different so there's actually multiple kinds of nematocysts within these lineages. So there's batteries. So as you go across those long hanging tentacles that can extend meters underneath the pneumatophore, there are different tentacles that can be specialized on feeding on different prey items, like different taxa. Isn't that amazing? That's kind of, it's like having a quiver of your harpoons that are attached to your arms that are floating underneath the water. They can also be bioluminescent. And so if you've seen a sailed jelly like this, it was a hydrozoan. And if you bump into them underneath the water with some of that, the Subastian and the remotely operated vehicles that are kind of sending back the high resolution video, I posted something on Blue Sky yesterday because even just as the initial drop of the ROV was happening, they bumped into 3 different phyla. They bumped into a ctenophore, and they bumped into a crustacean, so an arthropod, and they bumped into a gorgeous siphonophore that looked like this one here. Very grossly. I don't know if it was this species at all. But this colony that we kind of drill into the example of Physalia physalis as a siphonophore, a colonial that's got the pneumatophore that keeps it up at the surface. But there are lots of others of them through the water column. All right. How do they process information from the eye? Yeah, that's a great question. And we're 3,700. Yeah, take 3,700. This is a great question. All right. The last of these taxa that we're going to talk about today within the Cnidaria, sad face, is the myxozoa. And you can see, and you remember, what the dashed line, I suppose I should have taken some, the dashed line means uncertainty in this phylogeny. That might have been kind of implicit to what I was saying earlier about the Polypodozoa, but the dashed line means we don't really know where it's going. This is our hypothesis, and we don't know some things to characterize it. But synapomorphies of the Myxozoa seem to be associated with extreme reduction in size, extreme loss of all of the other characteristics, many of the characteristics of the synapomorphies that characterize the other Cnidarian lineages. Such simplification that up until 2015, we thought they were a phylum of protozoan. We've touched on them in this course very briefly when I took this course in the last century. This was a protozoan group, and it was very associated with the apicomplexans because one of the features of the Cnidaria that they retain is a nematocyst, a stinging cell, that looks a bit like an apical complex. Remember at the top of the plasmodium or the toxoplasma, that little bit that helps break into the cell and helps the protozoan kind of sneak into the host cell. So until 10 years ago, they were their own phylum, until we started to be able to collect them enough and to sequence them. They look, I don't know, creepy to me. But there's, I would say, the top, because if you flip them on, they look like an arrangement of organisms, but including kind of alien-style skulls. The movie Alien or Alien-style skulls in that SEM. But at any rate, as soon as in red here, if you see the reds, this is where they're sitting. So as a sister group in this analysis to the Scyphozoans, like an early branching lineage from the Medusazoas. So we've got anthozoans, the corals, and then we've got a lineage that contains the parasitic Myxozoans and all the medusoid forms. So an enormous reduction, which is kind of bananas. So this, what we were calling a polar capsule, and we still have called it a polar capsule as we've moved it phylogenetically. This is essentially an adaptation of the nematocyst. So they are cnidarians, not parasitic cnidarians, not parasitic protists. And so instead of being excited about the creation of a new taxon, as we've done with naming species and that kind of thing, this was a moment, a decade year ago when we said goodbye to a phylum. And revisit this large Keeling and Friends phylogeny. We had put it with the apicomplexans, and so this is a long, torturous taxonomic journey to say we thought it was this. It is, in fact, that. And as we learn more about myxozoans, they're weird. And the process of losing characteristic features extends. This is a proceeding of the National Academy of PNAS paper that includes some authors from Canada and was reviewed by Ford Doolittle, who is a Canadian biologist at Dalhousie, in February of 2020, where the cnidarian parasite, the myxozoan parasite of salmon, and we know about this one again because there's classic taxa that can deal with us, that either we know a bunch about a lineage because we can culture and cultivate one, or because it affects agriculture or aquaculture. So we know about this myxozoan because it affects salmon, aquaculture, salmon we grow in aquaculture. They don't have a mitochondria. So dial it back and not say we've lost a whole bunch of things that characterize the cnidarians. This is one of the characteristics of being a metazoan. So this is a real stripped down view of how to live as a metazoan or how to live as a cnidarian. You are a parasitic myxozoan, and this is one of the groups that as I teach this course through time and generations, this is one that we're learning amongst the most about. And so I anticipate that if you wanted to come back and teach it or go into cnidarian research, this is one where we have a lot to learn. Oh boy, so much to learn. So, two important things here. This is where we've been. And we've talked about the ecology, who eats whom. We've talked about all these elements of their movement, about their phylogenetic systematics, the diversity, misunderstandings that are recalibrated. We're going to stick next week. We're going to start out sticking with gelatinous things and then move to flattened things for the second part of the week as we move into the bilateria. But the small announcement at the end is that we have next week, all of the lectures are going to be like Wednesdays because I have been called away to a thing, a board meeting that I have to attend. And so we're going to be talking about ctenophores on Monday. On Wednesday, we're going to be talking about Xenocoelomorpha. And on Friday, we will be talking about the platyhelminth flatworms. And I hope when you share these enthusiasms for the ctenophores and the xenoturbellans with me, that you become as excited about them as you were about the arthropods and the cnidarians. Now, what to do over the weekend? You've got the quiz from this week from 789, lecture 789. So sponges, placozoans, and cnidarians. And that'll be due next Tuesday. Because I won't see you, I'm going to remind. So then there's the fourth quiz will be due kind of the Tuesday after that. And you're on to the schedule for your second week lab. So before you come to your lab next week, please complete the online tutorial for the microscope and make sure that you read about the lab, in the lab notebook, read about your second lab. And over the weekend, it's going to be gorgeous, like a good winter weekend. So do some good things for yourself. Take care of you, take care of those around you, replenish and refill whatever gives you energy. And I will see you virtually next week talking about ctenophores, xenoturbellans. I'll stop recording in a second and answer any questions if you've got them as you can before you head to your next class. So be good to you, be good to those around you, and talk to you soon.